Okay, so we'll finally make our way through the cell membrane. This will be our last big topic coming through is the active transport. Then we'll get into a little bit of the other active processes, endo and exocytosis. And then we get to dive inside the cell finally, right? Get into the cytoplasm. The organelles are the easy part of the chapter. And then lastly, we'll do some mitosis and protein synthesis at the end of the chapter. So as of right now, I realize the test is scheduled
for Friday.
And she's been doing some praying, man.
Yeah, she has.
This girl, she controls the universe, it seems. I love that prayer. It's probably going to be that Monday, okay? Look at that smile. I love it. All right. Well, anyway, so from this picture, this is like a static shot of the sodium-potassium pump. What do we notice, first of all? We notice it's a membrane protein, of course, right? That means it's called a protein pump. I don't want to get you confused on that terminology. It doesn't pump proteins. It's made of protein. So when I say a protein pump, know that it's made of protein, but that it pumps what? The ions, right? It's this flux of ions that gives the cell membrane its membrane potential. So it's the very heart of what we call the resting membrane potential. And based on this shot right here, the activity is telling us that it's pumping sodium out and then pumping potassium in. So we could take that and run and say, what is the major extracellular cation? Sodium. What is the major intracellular cation? Potassium. Okay. So let's just, hopefully if Microsoft Word, or Internet Explorer comes up fast enough we can see this in action. This is one of the videos posted on the YouTube channel.
Your Wi-Fi is down too.
What's that?
The Wi-Fi here is down too.
I don't think so.
Oh. Never mind.
It could go down any second. There we go. There's our sodiums going out. And that same loose phosphate from the ATP molecule is still fueling it. And it'll fuel the two potassiums coming in. Notice the detachment of the phosphate. And then a new ATP molecule will need to come in to energize it for another cycle. Okay. So over and over and over and over again. You get the point, right? Just keeps doing it. What it's not showing you is the passive transport side of things. But even again, in this static shot, what do you see? You see sodiums where? Primarily, you know that they're on both sides, of course, but they're making it a point to see easily here. Sodiums are going to be more so outside the cell, and potassiums are going to be more so inside the cell because of the nature of the pump, right? So let that, if you're a visual learner, let this be maybe the easiest way to get it instead of memorizing it. But it is 3 sodiums out for every 2 potassiums in. What else do you see inside the cell? A phosphate, right? So now we take it and run with that. We said the major extracellular cation was sodium. We said the major intracellular cation was potassium. What's the major intracellular anion? It's the phosphate. And proteins. Proteins are not seen here, but there are proteins always inside the cell, large proteins that are generally just trapped in the cell because they're so large.
That was the intracellular anion.
Anion, right. Is phosphates and proteins. So we're missing one little piece of information. What is the extracellular, the major extracellular anion? What usually goes along with sodium? Chloride. That's right. So we don't see it in the picture, but that's going to be our major extracellular anion. So let's get a quick sports update, recap on our ion teams, right? So extracellularly speaking, that's a very tough term to say. Holy cow, look at the snow. That's awesome.
Was it an option?
That's beautiful. Okay. Extracellularly speaking. Say that with me, guys. Extracellularly speaking. Man, that's tough, especially when you're cold. The major cation outside the cell is sodium. The major anion outside the cell, chloride. There are other anions, other cations, but these are the major ones, which means they're the most highest concentration outside the cell. On the inside of the cell, major intracellular cation, potassium, major intracellular anion, phosphates and proteins. Great. Okay.
Can you say it again? You confused the cat out of me just now.
Okay, I do that a lot. On the out, make yourself two columns, extracellular and then intracellular. Extracellular and intracellular. And then put yourself on the left, cation and then anion, right? So this is like another study tool that you guys could use. It's just one, one question. But an easy way to, people ask me, how do you study for this stuff? The way not to study is to always just read your notes and flip through your PowerPoints and nod with agreement. That's one way, right? But that's not the only way you should study. You should always kind of own up to it and write some stuff down, which means when you study something like this, take it and then write it down. Don't just read it. So we have extracellular category and intracellular category, just with the writing on top. Then make yourself the word cation and then anion, something like that, right? So we just fill in the blanks. What is the major extracellular cation? Sodium. The major extracellular anion? Chloride. Intracellular cation? Potassium. And then here the anion intracellular? Phosphates and proteins. Okay. Make sense? Okay, so if we use this, and I'll go ahead and draw another picture. I need some new markers, guys. 1 second.
Are we still having a quiz?
Oh, I got plenty. What's that?
Are we still having our quiz in lab?
Yes. Unless unless they start canceling school, which is possible. They'll wait till it's at the last worst possible moment for traffic.
Wait till you have to wreck and
Yeah.
get home.
Hopefully that doesn't happen. Okay, this is another thing. We mentioned the three main things, and this will be the last big topic, right, last time I'll share this regarding this topic, because I want you guys to just take it on yourself and own it. We mentioned three things, right? Three things that drive the resting membrane potential. And the nature of the resting membrane potential is such that we have a net negative charge on the inside of the cell membrane. So when we say resting membrane potential, all that really means is that the cell membrane has the potential for action. They call it resting because the cell is not acting on an impulse yet. We all know the cell is never resting. There's always movement, but at least in this term of acting on an impulse, it's not acting on an impulse yet. But it has the potential to act on an impulse, so we call it the resting membrane potential. And The potential also refers to the charge that's created, the polarity on either side. So what's the charge on the inside of the cell membrane? Negative. That's what's important, right? If you want to follow the logic, when it's acting on an impulse, what do you think the charge will be on the inside? Positive. Let's just make it up as we go along, right? Sounds good. Okay, and it is positive. When we act on an impulse, the charge on the inside changes to positive. Therefore, we always know resting on the inside is negative. Action is always positive on the inside of the membrane. So it switches like a light switch. So what are the three properties or characteristics or actions that give us the net negative charge on the inside of the cell membrane? The first one is active transport, the first event. We could say the second one is passive transport. And the third, large anions, right? And then we'll fill in the blanks as we go along. So this is the example I'm trying to give to you. Don't just study this, memorize it, and then wait for it on the tests. Study it, memorize it, and then reproduce it yourself before you get to the test, which means take a blank sheet of paper, write out this list, write everything you see, draw this cell, and show me everything you can. Right? That way, act like you're supposed to teach it. That's when you know you really know it. To be honest, I think it's hilarious that I am a teacher, because I remember not knowing this stuff for a very long time. And I think it wasn't really important to me until I had to teach it. You know how it is. Because trust me, I never use this in my practice. Not once. All right, so if we fill in the blanks here, let's make ourselves a membrane protein, our sodium-potassium pump. And we know that it's going to pump, how is it going to pump sodium? Outside the cell. 3 sodiums out, and then 2 potassiums in, right? And we talked last time about anytime you pump 3 positive ions out and only 2 positive ions back in, that builds a deficiency, right? If you take $3 out of your account, only put 2 back in, and you keep doing that, you're eventually going to go negative, right? Right. Have we done that before? So there's our net negative charge on the inside of the cell membrane because of active transport. Right? Check on that one. That's one qualification for giving us a negative charge on the inside. Based on the deficiency of positive ions, only 2 coming back in when there's 3 going out. Just based on charge. Okay, and where's sodium high now? On the outside. So passive transport dictates ions move from high concentration to low concentration. So sodium will leak down its own gradient from high to low through its own ion channel, right? Which is going to be facilitated diffusion, right? How about potassium? Where is it high? Inside, so it's going to leak down its own gradient from high to low, passively. But we mentioned way back that the cell membrane is selectively permeable. In this case, it's more permeable to potassium for some odd reason, right? So when it comes to resting membrane potential, it has more to do with potassium, right? Remember that. When it comes to action potential, which we're going to do in another chapter, it'll be more dependent on sodium. So that's another word association, or term association. When you think RMP, it's all about potassium. When you think AP for action potential, it's all about sodium. Try to stick that in your back pocket until later on. So now if we fill in the blanks, we do this. 3 sodium out, 2 potassium in. What's the hallmark about passive? It's more permeable to potassium. Now that we've got more positive ions leaving because it's more leaky for potassium, that gives us another check, another reason to believe that there's going to be a net negative charge on the inside cell membrane because more ions are moving out. Okay? They're leaking out. All these reasons give us a polarity on the cell membrane. If it's negative on the inside, what is it on the outside? Positive, right? So this is the electrical charge. It's the nature of the cell membrane to be polarized just like that. What that gives us is the perfect, platform for an action potential to come along. So what we'll see in a later chapter is that a neuron's going to come up, right? This is the edge of a neuron. And it seems like neurons touch cells and tissues, right? You've seen pictures of nerves coming in. But when you magnify that touching point, there's no touching, right? We mentioned this before. There's always, looks like there's touching, but there's not. There's always a space. So here's the space. So we want to send an electrical message down this axon to get to that cell, to make it do something. Long story short, because I don't want to go into the whole thing right now because it's not on this test, all we're going to do is change this negative charge on the inside momentarily to positive. See the difference? And that little switch, that little spark of a change is what we call an action potential, a depolarization. What does depolarize mean? It means to take that polarization and reverse it, right? That's going to act like a light switch, like a blinking light. It's going to go all the way across the cell and excite that cell and change the way it behaves. So that's what's coming up later, right? This part's not on the test. This is. The last thing that gives us our negative charge is our large anions trapped in, right? So how do you draw tertiary proteins? Just make something up, right? They're very large, they're very squiggly, they're negatively charged. They're so big they can't necessarily squeeze out of these channels. They will leave the cell or be embedded in the membrane, as dictated by the Golgi. But in a sense, there's so many of them, they're trapped in the cell. They don't leave just by themselves very quickly like other ions might. So they're negatively charged anions, right? And they sit inside the cell membrane, giving another reason, our third check mark, as to why we have that net negative charge. Okay. So now we've taken a topic that sounds pretty crazy on paper. We see it fold out like this. Does it make sense? It's not bad, right? It may take a little bit of practice writing it down on your own. I don't know. But at least you know it's possible. It's not something that's impossible.
So are you
Oh,
saying
thank
that
you.
finding on the
Thank
test
you.
that we're gonna have to label the cell?
I'm saying this is the way you should study it. I'm not going to have you label something on the test, no.
Okay.
But it's best if you could. Because you've already gotten a glimpse of some of my questions. Some of the questions are very detailed, And I'm not doing it like some people say my tests are hard. I really don't care, It sounds cocky to say that. But do you really, should the test be that easy so that you get to nursing school and you fail? No. Right?
What if you don't want to be a nurse, though? I'm just saying to get this grade.
Well, I know. I hear you on that.
I don't necessarily agree about being a nurse.
But this class is geared towards people going toward allied health.
I know, but I'm not going towards y'all career. So
What can I say? You just got to tough it out and smile, which you're doing. You're really good
trying
at
to
that.
do that.
I feel you. I do. I had to learn how to deliver a baby, and I never wanted to deliver a baby. So sometimes we have to learn stuff, But yeah, it is tough. And I do feel for you guys, I'm not trying to make the test hard. I just have to challenge you. I have to make you think, I have to make your brain grow, or your grain bro, whichever one makes you happy. Yeah, and this is the way to do it. And it's not like all questions are tough, but some questions have to be tough. Okay, so moving on. These are all the qualities here of active transport for the sodium-potassium pump against the gradient. If we look at vesicular transport, this is a small animation here showing you how the cell can move things inside of itself. Did we already watch that video of the bacteria? No? Let's see that real fast. Think about this cell right here. If it were a white blood cell, and if it were chasing a bacteria and then gobbling it in, right? It's bringing something inside of itself. You can see how it does it. The cell membrane folds upon itself, right? And it creates this little circle around whatever it brings in. That's called a vesicle. So what you see right here is a vesicle. It's really just a little chunk of the cell membrane that's pinched off. As something comes in, it swallows it with the cell membrane and then surrounds it with this vesicle. Because we don't want to just bring something inside the cell and have it loosely, being able to damage the cell. We coat it in this vesicle. So anytime we bring something in the cell, it's called endocytosis. Anything pushed out of the cell is called exocytosis. And when you look at the word, cyto means cell, osis means condition of, exo means exiting, right? Or endo means bringing in. So it's a condition of bringing something inside the cell for endo, or a condition of pushing something out of the cell for exo. Transcytosis just describes it moving across the cell, transporting across the cell in its entirety. Don't worry about the trafficking. So let's look at another slide real fast. On the bottom, the endocytic cycle here. Particles being brought inside the cell. You're seeing them being wrapped by a vesicle, and then they're carried inside the cell. If the cell wants to push something out, that could be a protein, that little red dot. It wants to push the protein out. There we go. It pushes it out. This could be a hormone, an enzyme, a mucus droplet, a neurotransmitter, all different methods of exocytosis. Anytime something is pushed out of the cell like this, using the cell membrane. Notice we're not using diffusion, right? There's a whole different kind of method here. We're not using a protein channel. We're using really the entire structure of the cell membrane to push something out. So it's different. All right, so let's go to the YouTube channel real fast and watch that video. If you haven't found it yet, it's always under Course Home. And just under Chapter 3, you can search for it in the playlists, and it's, where'd it go? There we go.
In this spread, a neutrophil is seen in the midst of red.
That's a white blood cell, the big one.
Staphylococcus
These are red blood
aureus
cells.
bacteria at the end.
There's a small piece of bacteria.
The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Okay. I never noticed that before. I've never had the volume turned on. That's pretty neat. Okay, so they call that phagocytosis because it's ingesting a solid. So there's our term right there. Anytime the cell ingests solid material, it's called phagocytosis. Anytime you ingest a liquid droplet, it's called penocytosis. Think of Pinot Grigio or something like that for liquid. Okay, so if we go back to this picture here, there's a sequence of pictures. What would you call this event right up here at the very top, number 1? It's coming in, so it's endocytosis. As it's moving across the cell, called transcytosis. Then we meet up with the lysosome. Lysosome does what? Digests whatever it comes in contact with, right? And then we spit out the waste. What's that event called? Exocytosis. Beautiful. So do we need to learn all this stuff? Nah. It's nice to read it because we know what some of it means, right? But just know the three main events. See, that part was easy, right? I'll give you a break every now and then.
Yeah, then you see your test, it's like, what?
I know.
70 questions in 45 minutes, that's awesome. Gotta love that.
That's right. Great for the brain. All right, what about on this? Are these Swedish fish? Or are they membrane proteins? Right here, right? Notice this is called receptor-mediated, which means we don't let something inside the cell unless it matches that receptor, that purple protein, right? Embedded in there. So notice this right here is something that matches the shape of that receptor quite nicely, which means it activates the whole method of endocytosis. That's why it's called receptor-mediated, because that receptor has to have its needs met before it allows something to come in. This is, in a sense, how a medication or a drug would work. A drug could come in and mimic the shape of whatever that is, steal all the binding sites on that receptor, and dictate how that cell is to be, functioning. So then it blocks the actual real thing that should be coming along. So drugs are kind of bullyish, truthfully. They kind of direct physiology in their own kind of way, which is good. But also realize a lot of medications don't actually heal. They just kind of trick the body momentarily, right? And that's not a bad thing. It's just sometimes we need to be tricked. Which you got to understand in a whole body sense, in a healthy sense, there's more to be done than just take medication, right? We got to take responsibility for our body. We got to eat better, exercise, that kind of thing. So realize that drugs aren't always the answer because of that reason. Okay, another view of exocytosis. You can see how the cell membrane just blends together and then pushes the product out. This could be a mucus droplet or a neurotransmitter release, like I said. So know all of these examples here. Hormone secretion, neurotransmitter, mucus, and wastes being ejected. Everything I've mentioned here, the old lecture right now, has all been not active transport, but an active process, right? None of this was passive. None of this was diffusion, right? We talked about the sodium-potassium pump primarily over there on the cell model. We mentioned passive transport in response to the ions leaking across their gradients. But even this right here, endo and exocytosis, is considered an active process. Okay. All right, one last review of resting membrane potential. I want you guys to know it has a range from -50 to -100 millivolts as the amplitude. I would know this as just -70. Let's just make it easy. The resting membrane potential measures at -70 millivolts. 70. Yeah, -70. -70, 70. And we already know this, right? If I could turn this on, there we go. The 1, 2, 3, same as what's on the board, right? Can you write it out? If I gave you a blank sheet of paper right now, could you write it out? In your own words even, right? Doesn't have to be my words, but it has to be correct, You can make up your own words, but it still's got to be correct. All right. 4, 5, and 6 here, I'm not as concerned about. We can ignore this part. Just know the 1, 2, 3. Here's another static picture of what's going on. We get to see these large protein anions on the inside of the cell. Notice if you take a picture like this, you can pull out quite a few test questions, can't you? If this is supposed to represent the resting membrane potential, you can see the placement of the sodium, right, with the chloride. You can see how potassium is moving, right? And you can see the protein anions trapped inside the cell. You see the negative charge on the inside, the positive charge on the outside. So lots of things can be said about that. Okay, the last little piece of the cell membrane is going to be cell extensions, but before we do that, any questions on our we're finished with transport now, right? So pat yourself on the back. When I say we're finished with transport, I mean, do you have a healthy understanding of the difference between simple and facilitated passive? Have you got that yet? Do you understand osmosis? Could you write something down about it? Or could you speak to someone about it? Could you teach someone about it? That's how you know when you got it, right? Something, anything. It doesn't have to be the whole story, but just something. Because then it builds into something else. Having a tiny bit of knowledge about something is extremely powerful. That's why on an exam, you're supposed to work the questions that you know first. Then come to the ones that you're not so strong on. Do those last, because a lot of times you'll find information in the easier questions that you can use on the harder questions. Okay. So the last part of transport was the active stuff, right? I think we tackled that pretty successfully today. So cell extensions, realize the only ones I need you to know are going to be cilia and microvilli. Don't worry about the flagella. We'll get that in AMP2 on reproductive system. But cilia and microvilli are the two I'd like for you to know. And they have two purposes. The cilia are going to move substances across that cell surface like mucus in the respiratory epithelium. We saw this a little bit in lab with our cystic fibrosis case study. So if we take a look at it, they look like little hairs, don't they? Or actually large hairs on top of the cell. But if you look closely, it's not just a hair, it's actually a projection of the cell membrane coming up and then coming down again. Coming up and then down again, right? So this is the cell membrane actually coming up and all the way down over and over and over again. It's not just a hair like you would think. It's actually just a tall finger of the cell membrane. You could think of it like that. But in this case, it pushes mucus across the cell surface. The top of the cell is going to be called the apical surface. If we look at another shot here of microvilli, same idea. These are smaller. Notice again the microvilli on the top of the cell. This is called the apical surface. The bottom of the cell is called the basal surface. This is the epithelial lining on the intestines. The microvilli, its job is for absorption of nutrients. Absorption. The cilia, what was the job for the cilia? To move things across the surface of the cell, right? Microvilli, its job is for absorption. So in your small intestine, this is how you absorb nutrients. Through those microvilli. And again, they're small extensions of the cell membrane. Here's a blown up picture. You can see now has some actin filaments giving it a little bit of structure and strength to stand upright. What are those actin filaments made of? What's the P word? The P word that we've said 3,000 times. Proteins, right. Yeah. Proteins are everywhere. And where do we get proteins from? What's the life story of a protein in 4 steps? 4 easy kindergarten steps. Where does the life story of a protein start? In the nucleus. Then it goes to the rough ER, the ribosomes, yeah. Then it goes to the Golgi. Then it goes to the cell membrane to either be pushed out, like we saw in exocytosis, or to live in the cell membrane as a membrane protein, right? So again, the 4 major steps. nucleus for the DNA instructions, sending it to the rough ER in the cytoplasm to be produced as a protein, right? Then sent to the Golgi to be packaged and processed, you could say, like a plant manager, and then the cell membrane. Okay, so that brings us actually nicely to the different parts of the cell. We'll just do a little bit of this and finish with it. So we're done with the cell membrane now. Now we get to dive in. The purposes for my class here in lecture will be for you to know the function of each of these organelles. For a lab, you have to know the function and what they look like. All right? So if you missed lab this week or will miss lab from the weather, realize I've got videos of the cell model, the same one we have in lab, on the YouTube channel. And it's just as simple as watching the video and as a teacher pointing out every structure of the cell, basically giving you the instruction. You know, so it's not like if you missed lab last week or whatever, or can't go to open lab this week, that's the same thing, right? Same thing with the tissues. I have a tissue video that shows you all the tissues. Okay, so let's do this real fast. Where's my stick? All right, so what's this? Nucleus. On the outside, it's called the nuclear envelope. Notice the outer layer. It's 2 layers. The outer layer is continuous with the rough ER. That's great. But on the inside of the nucleus, we have chromatin, which is a loose form of DNA. On the inside of that, in the middle, nucleolus. Inside of the nucleolus is the ribosomes on the inside. So there's 4 places we can see ribosomes. One of them is inside here. Another one is on the envelope. Another one on the rough ER. And then also freely floating ribosomes. Okay, so what's the whole function of the rough ER? Not only protein synthesis, but also the presence of ribosomes, but transport. Since it's so big, it's all the way across the cell. Think of it like an assembly line. It produces proteins and then transports them across the cell. Sends them to the Golgi, like we said before. What's the Golgi do? Packages, right, proteins, and dictates how they will be used in the cell or exited the cell. How about this one right here? Mitochondria in the orange. And its purpose is to produce ATP, right? It's called the powerhouse of the cell. The Krebs cycle takes place in here, if you've heard of that. How about here? TCA, yep. Centrioles, important for mitosis. It directs cell division using these microtubules. These aren't light rays. Well, actually they are. Everything is light rays. But they're microtubules. They're protein filaments that keep the chromosomes in place, right? Over here is the lysosome, important for digestion. What about these red filaments here? Perfect. Cytoskeletal structural filaments for the cell. Keeps the shape of the cell. How about up here? We missed that one. Smooth ER, lipid and carb transport. Okay, guys, so that's a whole chunk of the chapter out of the way now. We'll start with mitosis next time and protein synthesis.